Hi everyone. Yes, I'm really excited to be here, and thanks so much for having me and for the kind introduction. It's difficult to live up to that sort of introduction. But so what I want to do to you today is basically I just want to talk to you about social science genomics and how I'm thinking about it. And there are a couple of bases I want to touch. The first thing I want to talk about is why, from my perspective, social scientists should actually be really excited about genetics. And then I'll talk about some of the methodological challenges that we have to deal with when we want to identify specific genetic markers or genes associated with behavioral outcomes, and how we can actually address these challenges. And so the first part of my talk is going to be more theoretical, conceptual. It's going to be a little bit statistics. And then at the end, I will show you some empirical examples that are just going to put some numbers and give you better feel of where we are in the field right now. So I'd say it would be really great if we could just have this as a discussion. So whenever you guys feel like you have a question or you want to jump in, please feel encouraged to do so. And I can always cut some stuff at the end from the empirical examples if we run out of time, but I'd prefer to just have a discussion with you, okay? All right, so before I go on, I just want to give a little shout out to the many, many collaborators that I have. And most importantly, it's my amazing team that I have in Amsterdam. So these are the folks on the left. But I've also been working with a lot of people from a lot of fields from around the globe, and I just want to point out a few of them specifically. So Dan Benjamin and David Cesarini, these are the two guys that joined me in starting the Social Science Genetic Association Consortium, and we've done a lot of work together. And we've got a lot of support and also a lot of really valuable intellectual contribution from Peter Fisher, who is one of the leading genetic statisticians. So the list of collaborators goes on and on and on. I just want to make sure that you guys understand some of the stuff that I'm talking here about actually has been done by my collaborators. Okay, so why should social scientists be excited about genetics? Well, the first reason is that actually genetic influences are everywhere. Eric Turkheimer wrote an influential review article about behavior genetics in, I think, 2000, called The 3 Laws of Behavior Genetics. And the first law is that all human behavioral traits are partially heritable. So that seems like a very bold claim, but it actually turns out that this is supported by a really broad body of research. And primarily the way that people have studied that in the last 30 or 40 years was using the classic twin study design, where you basically use monozygotic and dizygotic twins as a naturally occurring experiment where you say, well, if we assume that these two types of twin pairs, that they share their family environment to the same extent, but we know that the monozygotic twins are genetically identical, but the dizygotic twins are only sharing 50% of their genome. We can use that to basically do a variance decomposition of any trait that we're interested in and just back out an estimate of how much of the total variation of the trait can be attributed to genetic differences among people versus shared environment versus individual environment. So that was the traditional paradigm for estimating heritability. And my collaborators, my colleagues at the Vrije University in Amsterdam, they published a nice paper together with Peter Fisher in 2015, which was basically a meta-analysis of all these twin studies that had been done up to 2014, I think. So they looked at this entire body of literature, which was almost 18,000 traits that have been studied in almost 3,000 publications based on over 14 million partly dependent twin pairs. So there's a massive amount of data. And basically what they found is that for all the categories, for all the phenotypes that they looked at, they basically rejected the null hypothesis of no heritability. And of course, they found a wide range of heritability estimates, and across the various traits, the average heritability estimate was pretty much exactly 50%. So let's talk a little bit about what heritability means. So this is actually conceptually important because it will also help us to understand what genetic associations mean and what they do not mean. So that's quite important. So one of the unfortunate things that happened in the last couple of decades is that all these heritability studies were very often framed in the context of a debate about whether things are nature or nurture. I think nowadays in the behavioral genetics community, there's a very broad-ranging consensus that this debate was actually conceptually flawed, because it is neither only nature or nurture. It is always an interplay of the two. And very importantly, the estimates that we're getting from all these various statistical methods, not only the twin method, but also methods that use genome-wide data, the estimates that we're getting, they're dependent on the environments in which people are being sampled. They depend on the specific sample that we're selecting. So the modern consensus is actually that most traits result from a very complex interplay of nature and the environment, rather than either one of them separately or isolated from the other. So heritability technically just refers to the proportion of observed differences in a trait among the individuals of a population that is due to genetic differences among these individuals. And just to clear up some of the very persistent misunderstandings about heritability. So I know that a lot of people feel they feel uncomfortable whenever you mention genetics or heritability in the context of human behavior or socioeconomic outcomes and things like that. And primarily it's because there is this idea that when something is heritable, that there is some biological determinism, but that is actually a conceptual misunderstanding. So heritability does not put an upper bound on the potential relevance of the environment. And this is even true if you have a trait that has a heritability of 100%. You can show that mathematically, but you can also show it with thought experiments. I'm going to talk about some of them in a minute. It's also important to understand that heritability is not a natural constant. It's nothing that is God-given, that drops from heaven and we just have to deal with it. It's rather a consequence of the environments that we live in and the constraints that we have. And as a result of that, you actually find that heritability estimates of the same trait often vary across environments and over time. So for example, you see pretty remarkable differences in the heritability of body height when you study populations in Western countries versus populations in sub-Saharan Africa. So the heritability of body height is substantially lower in sub-Saharan Africa for the very simple reason that body height is a function of not only your genetic endowment, but also access to sufficient nutrition and healthcare. And these are things that are basically universally accessible in the Western world. So in that case, the amount of the variation in the trait that is due to differences in access to these resources is restricted, and as a result of that, heritability goes up, right? So there is no biological determinism or absence of choice that comes with the heritability estimate. It may be the case that a high heritability actually restricts how much you can actually do about a particular outcome. But this is primarily a function of technological development. So I'm going to give you an example. Let's say you think about short-sightedness. So short-sightedness may actually be 100% heritable. But if you live in a society where glasses have been invented and you have access to them, it's very easy to fix, right? It's just it's a matter of just getting the glasses and putting them on your nose and you fix the problem. So for a lot of traits that have high heritability, there are these sorts of interventions already available, and for some there is not, but they may actually emerge in the future. So very important is that genes exert their influence very often through environmental channels. And we're picking up these environmental channels actually with these heritability estimates, right? So sometimes the effects or the heritability that we find, they are conditional on the environment. They may actually be induced by an environment. So I'm going to give you an extreme example to illustrate that. So let's say you have a dictator who decides that all boys who are born in that society have to go to the army, but they're not allowed to go to school. But all girls, they have to go to school, but they're not allowed to go to the army, right? So that's a completely ridiculous example. But in that society, if you would do a heritability of whether you have a schooling degree or whether you went to the army, the heritability would be exactly 100%. And furthermore, you could actually, you could do a genetic association study and you would find that, what matters is whether you have 2 X chromosomes or 1. But obviously, there is no meaningful biological mechanism that restricts girls from being able to go to the army or from boys being able to go to school. This is the type of genetic association or the type of heritability that would be entirely induced by an environment. Okay? So we need to keep that in mind. Also, in a lot of cases, the heritability actually plays out through self-selection of individuals into specific environments or through specific responses that people with a particular genotype evoke from their environment, right? So again, you may think of an extreme example. Let's say you live in a society in which, for whatever reason, people discriminate against people with red hair. So if you're born with red hair and the environment responds to that in that way, that, for example, you may not get a good job or you may not be accepted to the best colleges, then if you run a genome-wide association study on, let's say, income or college attainment, you would probably find an association between the gene that codes hair color and having a high income. But again, it would be entirely due to environmental circumstances. All right? So, and as a result of all of that, all of that makes it actually a bit difficult to interpret GWAS results or heritability estimates as clear evidence for anything. But very importantly, they're not informative about the nature of differences between groups or over time. Okay. I'm just going to show you a couple of heritability estimates that we're getting from Western populations that are typically being studied. So, of course, there are some traits that are basically entirely genetic. So these would be typically the monogenetic disorders that are very rare in a population that occur very rare and that are caused typically by just 1 or 2 very well-known genetic mutations that basically cause the defect. But then you have this vast array of traits that can be studied where we see a heritability, but the heritability is actually not due just a few genes with large effects, but they're rather what we call genetically complex traits where the heritability actually comes through hundreds, if not thousands of genes with tiny, tiny effects. So a good example of that is height. So like I said, height is very heritable in Western civilizations, and it's still it's a highly genetically complex trait that basically has been associated with thousands of genetic markers. So the things that we're interested in as social scientists, these are the traits like personality or socioeconomic outcomes, employment choices, so on and so forth. They typically have moderate heritabilities, so in the range of something between 30 and 50%. And then there are some outliers that seem to have very low heritability estimates, like risk preferences. And what's interesting about risk preference is it's a nice example for what happens when you measure a trait with a lot of noise. So if you have a very noisy measure of your outcome, that actually means it attenuates the heritability estimate because of noise is not heritable. So this is actually one of the examples where if you correct for measurement error, the heritability estimate actually goes up quite substantially. Okay, so what's in it for social science? So how can we possibly integrate genetic insights into our work, and why should we be excited about that? I think there are a number of reasons for that, and I think primarily from a social science perspective, the really cool thing is that genetic insights will ultimately help us to better understand the environment and policy effects. So there are a number of ways how we could actually benefit from that. And a lot of them, they have to do with polygenic scores that actually aggregate genetic signal across the entire genome and try to put a number on the overall genetic predisposition that an individual has to a particular outcome. So if you have good polygenic scores, we can use them for things like reducing unobserved heterogeneity in regression models, right? So we know that if there are unobserved confounds, they're gonna bias the estimates of the things that we are actually interested in. Now with genetic data, the cool thing is that as soon as you have genetic data, you can basically construct polygenic scores for all traits that were ever studied in a GWAS, which is literally thousands by this time. And you can, in principle, include all of them, or at least the relevant ones. You can include them as control variables in your regression, which is going to reduce bias from unobserved heterogeneity. It's also going to increase statistical power, because if you have a control variable that absorbs residual variation in your phenotype, it's basically going to give you smaller standard errors of the effects that you're actually interested in, which may be behavioral or policy exposures, right? Furthermore, we can then actually take the genotypes into account and actually investigate how they interact with specific features of the environment. And there are a number of new methods that are currently being developed which help us to use genetic information to some extent to get closer from just pure correlations towards causally informative estimates. And then there is another aspect of having genetic data or polygenic scores which just became clear relatively recently, and this is the idea that you can use these polygenic scores to study intergenerational transmission. So what we see is that typically for most traits that we're interested in as social scientists, such as socio-economic status or personality, there is quite a bit of persistence across generations, right? So you see that parents resemble their children, the parents resemble their parents, and so on and so forth. And the obvious question is then, so how does this transmission from one generation to the next work? Is it genetic transmission or is it environmental transmission? And in order to tease these two things apart, traditionally you needed to have adoption studies or you needed to have children of twin studies. And it was relatively difficult to get your hands on that. And it was actually either a very rare or a very invasive research design that also didn't really capture the entire spectrum of society that we would actually want to study. Now, with polygenic scores, you can actually do something super cool. So if you have genetic data for the children and their parents, you can basically estimate or you can put a number on the genetic endowment that the children have by just taking their genome. So that's the part that they basically got from their parents, 50% from the mother, 50% from the father. But then the mother and the father also have 50% of their genome that they did not pass on. And if you have measured them, you can also construct polygenic scores for the parts that the parents did not pass on. So now you have a natural experiment where you have a part of the genome of the parents that still influences what the type of environment that the parents create for their children, the parenting style, the socioeconomic status that they have, even the neighborhood that they live in. All of that will probably be associated with the part of the genome of the parents that hasn't been passed on. And now this is going to be orthogonal to the genes that have actually been passed on, and you can use that to identify causal effects, right? So you can then tease apart which parts of the parental environment actually mattered, and you can put a number on that. So I think this is actually pretty cool. Also, we can use the empirical results from genetically informed study designs to inform our theories in the social sciences. So one thing we can do is we can actually use genetic data to test concepts or ideas about the heterogeneity among people. So one example would be risk preferences. So economists like to talk about risk preferences. But there is a question about whether risk preferences are actually a real thing, or if it's just something that that the microeconomists came up in the middle of the night. Is it something that is situation specific, or is it something that is stable over time? And genetically informed study designs can actually help us to test some of these things. Another thing that is actually surprisingly useful and informative is our ability to actually test genetic correlations among variables. And we can now do that with the summary statistics from genome-wide association studies that are very often just publicly available. And there's a very powerful and easy method called bivariate LD score regression, which actually allows us to compute the genetic similarity among traits, even among traits that haven't been studied in the same set of individuals. So this is entirely new, right? So if you want to do a normal correlation, you actually need to measure both variables in the same set of individuals. With genetic correlations, you don't need to do that anymore. So you can run a genetic correlation of, let's say, IQ in children that has been measured in one sample with schizophrenia or Alzheimer's disease that has been studied in a completely different sample. So that's really cool. And furthermore, you can then also do the entire toolkit that is being used in psychology to also model multivariate relationships among these traits using only genetic data, either from overlapping or non-overlapping samples. So that opens up a lot of possibilities to test relationship and to actually see relationships for the first time that we were not aware of. And then there is a surprisingly large overlap between social science genomics and interests in furthering health research or genetic epidemiology. So the reason for that is that it turns out that a lot of the medically relevant traits or a lot of diseases, they actually have substantial genetic correlations with traits that we're interested in as social scientists, such as education attainment. So education attainment is actually one of these variables that is basically just correlated with everything, right? So it's robustly associated with health outcomes, with longevity, with basically everything. And it turns out that this is not only true at the phenotypic level, but also at the genetic level. There is a substantial genetic correlation between education attainment and a lot of health outcomes. So why is that important? Well, one of the reasons why that's important is that it basically enables us to use insights that we have from social science genetics to actually improve or to speed up medical research. And that works because some of these social scientific variables, they're super easy to study or to collect, right? So they basically education attainment basically boils down to just asking one question in a survey. So it's super easy compared to having, for example, a case control cohort or to conduct brain scans on people. Right, so it's very easy, it's cheap to measure. So you can get gigantic sample sizes, and very often these data have been collected, so you don't need to go out and collect them from scratch. They have already been collected, you just need to tap into them. And then you can use the GWAS results from these enormous sample sizes to actually inform research on medically related traits that often cannot be studied in such large samples yet. So there are a number of statistical techniques that you can use for that. Some of them have also been developed in our group, such as the proxy phenotype approach or the MTech method. There's also a lot of ways how you can actually model the multivariate genetic relationships between variables, so both including socioeconomic and medical outcomes. And it turns out that there are also a lot of examples where the insights from social science genomics are ultimately informative about the nosology or etiology of specific diseases. So yesterday I gave a talk that basically illustrated how we can use the GWAS results for education attainment to show that schizophrenia is actually a genetically heterogeneous outcome that can be decomposed into various subtypes that have different biological causes and that may actually constitute different diseases. And for etiology, I mean, there's also a lot of traits where you can use Mendelian randomization or other genetically informed study designs to actually test for causal mechanisms. A nice example was recently a study design that actually used the naturally occurring experiment, an educational reform in the United Kingdom. And the question was whether education actually has a causal effect on obesity. So they used that, an increase in compulsory schooling in the early 70s in Great Britain as a naturally occurring experiment to identify that causal effect. Turns out, yes, people that went longer to school actually have substantially lower BMIs. And then they combined that with genetic insights, and they basically showed that the people that benefited from that policy intervention most were actually people that had a high polygenic score for BMI. So people that were genetically especially predisposed to be affected by obesity were the people that benefited most from that policy intervention. So there's a lot of stuff that we can do with these type of data in the future. So why now? Why hasn't this been done already like 10, 20 years ago? Well, so integrating genetic insights into the social sciences requires at least two things. It requires, first of all, access to high-quality genetic data that simply wasn't available until very, very recently. But this has dramatically changed. So we're now living really in a world where genetic data, collecting genetic data is super cheap and it's abundant. So there's a lot of data out there that you can work with. The second thing is you need to have some sort of at least preliminary understanding of what I call the molecular genetic architecture of traits that are relevant for social scientists. So with that, I mean basically the joint distribution of the effect sizes and allele frequencies of the causal genetic variants. So this can be either simple or complex. For most behavioral traits, it's extremely complex. And so this part is still largely unknown for most of the traits that we're interested in as social scientists, but there is enormous progress being made at the moment. So there's literally on a day-by-day basis, there are new large-scale GWAS results being published that actually increase our insights about the genetic architecture of many, many traits. And until now, there is a really cool resource called the GWAS catalog, which basically has all the previously published GWAS findings in a huge database. So you can just go to the GWAS catalog, look up the trait that you're interested in, and it will point you to the studies that have been published on that particular trait and list all the genome-wide significant associations that were found for that. And literally, the GWAS catalog is like growing by the month. It's tremendously growing. There are thousands of replicable genetic associations for hundreds of outcomes that have been published in the last couple of years. So this is basically what's driving all of that. It's an extreme fall of genotyping costs over the last couple of years. So the first genome that was the first human genome that was sequenced actually cost around $100 million, and that was just in 2001. Now we're down to a full genome costing you not much more than $400 or $500 to completely sequence it with high accuracy. But the cool thing is you don't even need sequencing data. For most purposes that we're interested in as social scientists, what you need is basically just variation of common genetic markers, so genetic markers that are actually different among, let's say, at least 1% of the human population, right? So if you limit it to that, you don't need much more than about 1 million markers, and they can be collected in humans for as little as $20 nowadays. So it's ridiculously cheap, right? And the costs keep falling. So, okay, so let's talk a little bit about some of the methodological challenges. So just as a background, so as humans, we have roughly 3 billion base pairs of DNA. Those fall into, or there are roughly 22,000 genes. These genes, they code proteins or several proteins. And then across the entire DNA, we have various types of genetic variation. The working horse of all genetic work that we're doing at the moment are so-called single nucleotide polymorphisms. So these are basically just specific locations in the genome where some parts of the human population have a different base pair than other humans. So it's simply a variation of a base pair at a particular locus. And these single nucleotide polymorphisms, they are the most common form of genetic variation among humans, and they capture the vast majority of known genetic variation among people. And they have this property that you can just code them very simply as either 0, 1, or 2. So 0 means you basically have 2 copies of the reference allele, one from the father and one from the mother. 2 means you have 2 copies of the opposite allele, so the other possibility, one from the father, one from the mother. And a 1 means you're a heterozygote, so you have 1 copy each. And actually the assumption is that it doesn't matter if that copy comes from the mother or the father. There are other types of genetic variation that can actually matter quite a bit. So there are, for example, indels. So these are either insertions or deletions in the genome. There can be inversions. There can be all sorts of weird genetic variations going on. They're not captured very well yet with the genotyping arrays that we're currently using. But many of the SNPs actually turn out to be, at least to some extent, correlated with these structural variations that are not measured directly yet. Okay, so what do you have to do if you want to find out the genetic architecture of a trait? So let's say that the trait is a variable that we call y, and in order to identify the genetic architecture, this is basically the structural model. You would want to fit all the genetic markers that have been measured, so all these millions of markers, and basically just run a regression of y on all these genetic markers. You'd have an intercept term and an error term, obviously. We would now say that a genetic variant is causal if its true effect size beta is not 0. Okay? Now, one of the challenges here is that, as I just said, so you're going to have millions of genetic markers. So you're going to have, the J index is going to run in the millions. But you're only going to have n observations that you can regress on, and the n is typically going to be much smaller than a million. So if you want to estimate that equation with something like OLS, you basically have to worry about the basic assumptions that you're making in these statistical methods. So let's just walk quickly through what these assumptions are and what the challenges are. So the first one is that you actually cannot run a regression of your phenotype on all genetic markers at the same time, simply because you have too many of them. The equation is not specified. So what we do instead, and this is important to understand, is that instead of running one regression on all SNPs at the same time, we actually run one regression for each SNP. So we're literally running 1 million or over 1 million independent regressions, right? So that means that any correlation between the genetic markers is not taken into account, and as a result, all the beta estimates that we're getting, they're biased. To the very least extent, they're biased by the missing correlation of that SNP with other SNPs in the neighborhood. So this is also basically a characteristic of the human genome, that genetic markers that are physically close to each other, they tend to be correlated with each other, right? So we're not taking that into account when we run a genome-wide association study. Also, we're only measuring common genetic markers at the moment, which actually means that any genetic marker that is not measured at the moment, that is not correlated with any of the things that we're measuring, but still influences the trait, is going to be missing from the equation, and it's going to contribute to what we call the missing heritability problem, which is basically the difference between the variation of the trait that you can explain from genome-wide data versus the heritability estimate that you're getting from a twin study. Okay? So then the next obvious issue is that you're going to run a lot of independent hypotheses. So you have a huge multiple testing problem, right? So if you run a genome-wide association study on Europeans, you're testing roughly, it depends a bit on quality control characteristics that you impose and so on and so forth, but you're testing roughly 1 million independent hypotheses. So let's say you're running 1 million independent hypotheses, and actually there is no true genetic signal. So all the true betas are 0, right? And you have, let's say you use the classic p-value threshold of 5%. So you're running 1 million hypotheses. How many false positives do you expect to find? 50,000. So that's a little bit too much, right? So, I mean, you don't want to do that. I mean, of course, you could write 50,000 PhD theses with that, but I would really not recommend you to do that. So instead, what we do is we impose a multiple testing corrected p-value, which basically is taking the 5% threshold and dividing it by the number of independent hypotheses we're testing, which yields this famous number of 5 times 10 to the power of minus 8, which is what we call the level of genome-wide significance. So only genetic associations that have a p-value of 5 times 10 to the power of minus 8 or smaller are considered to be associated with that trait. Now, it turns out that as we get better genetic data, we may actually be testing more than 1 million hypotheses. And also there are some human populations that actually have much more genetic variation than we have as Europeans. And also in these populations, you may actually need to impose even more stringent p-value thresholds to correct for multiple hypothesis testing. Couple of other things. So when you run OLS, so one of the assumptions that you're making is that that every genetic marker just has a simple linear effect on the outcome. So that may actually not be true. So the effect may be nonlinear. For example, there may be a dominance effect. So in that case, OLS would still identify the causal genetic markers, but it would obviously not give you the correct beta estimate. But it turns out that for the vast majority of traits that we're interested in, the simple linear model seems to capture the heritability of the trait relatively well, so which seems to suggest that dominance or nonlinear effects in the genome are probably not something that we need to be too worried about. Also, there's going to be, I pointed that out earlier, so there's a possibility that a genotype may actually interact with various factors that are in the error term. So this may be either environmental or genetic effects that are, that are ignored because we're not running everything at the same time. So if that's the case, OLS will give us the weighted average effect of that X. So that's still useful as a starting point, because if we estimate that the beta of that genetic marker is not 0, this is a useful starting point for investigating if there is a G by E or a G by G effect. Okay. One thing we really have to worry about is the possibility that our genetic information is going to be systematically correlated with specific features of the environment which are actually causing the trait, right? So this would be a specific case of unobserved variable bias, where because we're not putting the environment into the regression, if there is an environmental effect that is correlated with the gene and that also is correlated with the outcome, it would lead to basically a spurious genetic association. So the famous example is the so-called chopstick gene, where let's say you run a GWAS on whether people eat their food with chopsticks or with forks and knives. If you run a GWAS on chopstick use in the American population, you would find a lot of genetic variants associated with chopstick use. But they would basically, all of them that you would find would actually have a difference in minor allele frequency between Asians and non-Asians in your sample. So basically, this would be the case where, whether you eat with chopsticks or not is obviously a cultural issue. So anything that is just correlated with being part of that culture, any genotype that is just different between that culture than the other cultures, would show up as a genetic association. So we need to address that issue, and it's actually, this is something that geneticists have thought about for a long time. They call that problem population stratification or population admixture. And there are various ways how we can address that. The most popular one is that we just we estimate the first few principal components from the genetic data, and we add them as control variables to our GWAS. So why does that help us? It's primarily because if you look at the first few principal components of the genetic data of humans, they correlate very well with genetic ancestry and with historical migration patterns, which are also correlated with a lot of things like where you live and which culture you grow up in. So it's a proxy, it's an indirect proxy that captures some of these environmental confounds. So it turns out that for a lot of medical traits, these first few principal components are pretty much effective in terms of getting rid of these chopstick genes and just getting rid of population structure. But there is no way, neither empirically nor theoretically, to actually show that you've done a sufficient job, right? So that by itself is not going to be enough. There are other ways how we can address that issue. So one of them would be linear and mixed models, where you actually fit. So you look at the genetic relatedness matrix of all the individuals in your sample, and you basically take that into account when you estimate the betas and the standard error. So that's a bit more rigorous in terms of addressing population structure. But actually, the gold standard for doing that would be if we would just do GWAS within families, right? So if we would just have a lot of siblings or a lot of dizygotic twins where we could just look at the genetic differences and the phenotypic differences between these twins or these siblings, then we would know that any genetic difference between them is completely random and is orthogonal to the environment that they grew up in because they share the environment. Now, so that would actually be a super convincing study design to get rid of environmental confounds. The problem is that for most genetic traits that we're interested in as social scientists, the effect sizes of the individual SNPs are so small that we just don't have enough twins in the world to do that, right? So the only thing we can do is we can use, these sorts of family fixed effects as a follow-up tool for all sorts of robustness checks later, but we still have to do the genetic discovery in population samples without family fixed effects in most cases. Okay, so then there is the big problem of small effect sizes. So most, if not all, heritable social scientific variables are likely to be genetically complex, which means that the heritability is going to be due to hundreds or thousands of genes with tiny, tiny effect sizes, right? So that ultimately means that we're going to run into a huge problem with statistical power, right? So statistical power, just as a reminder, is the probability of being able to reject the null hypothesis if it's actually false and the alternative is true. And statistical power is going to be low if your sample size is small, if the effect sizes are small, and your p-values are small. So in a genome-wide association study on something like education or personality, you're basically dealing with a combination of two of these problems, small effects and very small p-values. So what's the problem with running an underpowered statistical test? Well, there's two problems really. So the first one is, if you don't have enough power, the chance of you being able to identify true associations is very low. But the flip side is, and that's probably an even more serious issue that most people are not aware of, the flip side is, if you run an underpowered statistical test and you find something that's statistically significant, then you have dramatically overestimated the true effect of what you identified. And you may even get the sign of the effect wrong, right? Because your estimate is so incorrect, it's so far away from the true effect size. So in any case, if you run an underpowered statistical test, the results are not going to be meaningful because you're going to walk away with a completely distorted view of what the world looks like, right? So the bottom line is you should not run underpowered statistical tests, okay? So how do we deal with that in GWAS? Well, let's first have a look at what realistic effect sizes really look like. So this is a graph that shows you the relationship between effect sizes of a particular genetic marker in terms of R². So this is 0.1%, and this would be 1%. And the graph shows you all combinations of effect size and sample size that would give us 50% power at genome-wide significance. So where are we on this graph? First of all, you see, of course, that the required sample sizes, they basically exponentially increase the smaller the sample, the smaller the true effects are getting, right? So where are we on this graph? So if you look at Alzheimer's disease, well, we're actually up there, we're fine, right? So ApoE has a pretty strong effect, and people were able to identify it in a sample of just a few thousand individuals. Things look very different for things that we're interested in as social scientists. So probably an upper bound for how large plausible effect sizes could be are body height and BMI. So these are biologically pretty proximate or pretty there is a clear biological link between BMI or height and your DNA. And even for these traits, the largest effects that people have identified of a particular genetic marker are in the range of about 0.2, 0.3%, right? So that's a small effect. If you look at biologically more distal outcomes, such as what people do or what their socioeconomic status is or what their personality is, you end up, or this is a case here, this is psychiatric diseases, bipolar or schizophrenia, you end up with much, much smaller effect sizes. So you need much larger samples to identify them. And with education or IQ, you literally end up in a range where you need more than 100,000 individuals in order to have at least 50% power to identify these effects. 100,000 individuals, so that's a lot. How do we get them? Well, traditionally, the best way to do that was basically just to have a collaborative effort where you basically pool evidence from all the people around the world who have collected that data, let them run that analysis for you, share the summary statistics with you, and then meta-analyze. So that's the idea of all these genome-wide association consortia, and it's also the idea behind the consortium that we set up. And you could make a lot of progress with that approach for a lot of different traits. Now, recently, there have been a couple of new developments that have really completely changed the game, and that is basically the emergence of consumer genetics companies such as 23andMe that have collected genetic data from vast amounts of people. So 23andMe actually, it's super interesting what they're doing. So they basically convince their customers to pay them money to give them their data. So that's smart. And they managed to convince more than 2 million people to do that, right? And most of these people also consented to having their data being used for research purposes. So they're really sitting on a gold mine, and they can now run genome-wide association studies on millions of people for all the traits that they've measured in their surveys that, again, people volunteer to participate in, right? So that's a huge step forward, and a lot of the work that we're doing, a large part of the sample actually comes from 23andMe. But then there's also the advance of large-scale biobanks, such as the UK Biobank, where 500,000 Brits, a population sample, have been recruited to a medical center. They've been examined. They volunteered all sorts of information about themselves. Their data can also be linked to the digital health records of that country, and all 500,000 of them have been genotyped. And that data is in principle available for any bona fide researcher to access and to work with, right? So that's just amazing. So we're really now living in a world where, access to data or not having enough data is not that much of a problem anymore. It's rather, finding it and using it in a smart way. The other thing that you can do to address the power issue is, well, you can also think about more smart statistical approaches that are not as naive as the simple OLS approach that I was just showing you. And one way how you may actually gain statistical power is by taking into account information from genetically correlated traits, right? So there's a lot of methods that try to exploit that to gain statistical power. Okay. As social scientists, I think most of us are ultimately interested really in understanding the environment. And for us, the genetic endowment of people is either something that is a potential confound, and we want to make sure that our estimates are not confounded by it, or we actually want to understand which part of the population is actually being affected by a particular policy reform or something like that. So ultimately, we're more interested in the genetic endowment entirely, rather than whether people have one specific gene, or to understand what the biology of that gene is. So we can just summarize the overall genetic endowment by this index GI, which would just be a linear index of all the genetic markers that a human has, multiplied by the true effect, beta j, of those genetic markers. So if we would know all the genetic markers and we would know all the true betas, that would actually give us an alternative definition of heritability. So the variation in this index j, if you divide that by the variation in the phenotype, that would actually give us the heritability of the trait, right? So this would be what we call narrow sense heritability, and this would all be linear effects. Another way to think about that index j is this would actually be a polygenic score, so we could call it the true polygenic score. It would basically just aggregate across the entire genome using the true betas. So if we would know the true polygenic score and we would run a regression of y on that true polygenic score, then the r square of that regression equation would actually be the heritability of the trait. So that's another way to think about the polygenic score. So the polygenic score, ultimately, it can explain at maximum the amount of variation that is attributable to genetic differences, so the heritability of the trait, right? Now in practice, of course, we don't know the true betas. The only thing we have are estimates of them, right? So beta hat. So these estimates of beta, they're going to be noisy, and as a result of that, the polygenic scores that we can construct from these noisy betas is actually not going to have an R squared that is even remotely close to the true heritability of the trait, right? However, The thing is that our estimates of beta hat, they're going to get more and more accurate the larger our GWAS samples are getting, right? So this is an important thing. The larger the GWAS sample, the more predictive your polygenic score is going to be, and it's going to get closer and closer to capturing the overall heritability of the trait. You can actually work out what the relationship is between the expected accuracy of a polygenic score and features of the GWAS that you're running, such as the heritability of the trait, the GWAS sample size, and how genetically complex the trait is. It turns out that your polygenic score will be better if your GWAS sample is large, if your heritability is large, and if the complexity of the genetic complexity is low. Right? So the more complex the trait is, the more difficulty you will have to actually estimate the true betas. Okay, so all right, so let's stop here for a minute before I'm going to go to the empirical examples. Are you guys still with me? Do you have any questions, comments? All right, so then, again, feel free to interrupt me or to ask anytime, okay? So I'm just going to go on now, and I'm going to show you a couple of empirical examples from our work that are just going to put some numbers on these theoretical things that I just explained. So when we started out with our Social Science Genetic Association Consortium, so we set that up, I think, in 2010 or 2011. And this was in the middle of a time when people started to be interested in genetic associations with socioeconomic outcomes. But we were literally in a huge replication crisis because a lot of the work that had been done up till then has been done in very small samples where the effects that were estimated, like I said, so they were underpowered studies. The effect sizes were very, very far off from what the true effect sizes are. They often even had the wrong signs. And as a result, they also very often didn't replicate, right? So this was a huge crisis. So we said, okay, how about if we study a trait that we know is heritable, but we do it in a sample that is literally orders of magnitude larger than anything that has ever done before? If we don't find anything, that's still going to be interesting. So we set up a consortium with the goal to have at least 100,000 people where we can study socioeconomic outcomes. And the thing that basically everybody has collected was education attainment, right? So the most genetic data sets, they were collected for medical research purposes, and epidemiologists, they typically collect information about education, not because they're interested in education, but because they know that it's correlated with the stuff that they are interested in. Okay, so other people's leftovers are our treasures, so we just ask them to run GWAS on that. And assembled a sample size of over 100,000 people. Now, surprise, surprise, we actually did find something, and we tried really, really hard to make it go away, but it wouldn't. So we had 3 genome-wide significant associations, but the effect sizes of them, they were so small, they were orders of magnitude smaller than what previous findings in the Canada gene literature had suggested. So actually, the largest effect sizes we found, they were in the order of about 0.02% of the variation of years of schooling. So that was 2013, first time that this worked. 3 years later, we were able to basically triple the sample size. So we went from 100,000 to 300,000 individuals. And with a discovery sample of 300,000, we went from having 3 genetic loci associated with education to 74. And our polygenic score actually started to predict a substantial amount of variation. So in the first paper, the polygenic score captured about 2% of the variation. Now we were at about 4 or 5%. In that paper, so this was 2016, in Nature we had also a replication sample. So this was basically when the UK Biobank was released, the first batch of it, which was just a little bit more than 100,000 people. So we could use the UK Biobank as a replication sample. So for our 74 genome-wide significant markers, we're plotting here the effect sizes in terms of years of schooling in the discovery sample. So these are the black dots. And we're plotting them against the effect sizes that are estimated in the replication sample. These are the gray dots. And the bars around them are the 95% confidence intervals. So we see that from the 74 markers, actually 52 replicate at the 5% level, and 72 have the anticipated sign. And if you take into account the power that we had to replicate these effect sizes in this replication sample, you basically cannot reject the hypothesis that all of these associations are true. So this is a very, very strong replication record. So in the 2016 paper, this was also at a point where LD score regression had already been developed. So we were able now to use our GWAS results to look at genetic correlations of education attainment with other traits that have actually not been studied in the same sample. So one of the cool things is we could estimate genetic correlations between particular volumetric measures of the brain and education attainment. Turns out education attainment is genetically positively related to having a larger brain. Okay, maybe not surprising, but it's a pretty remarkable effect there. It's also related to various medical outcomes such as Alzheimer's disease or bipolar and schizophrenia. The strongest genetic association we find is with cognitive performance or IQ, that's about 0.7, but also with various measures of personality such as neuroticism, openness to experience, conscientiousness. And it turns out that these, you may call them endophenotypes, right? So these are personality characteristics that are sitting somewhere in the causal chain between the genotypes on the one end and education attainment on the other hand. So it turns out that the genetic effect is actually mediated by several of these personality measures, and these personality measures are not perfectly correlated genetically with themselves, right? So you should think of education attainment basically as an umbrella term that captures all sorts of endophenotypes that are actually different from each other in terms of their genetic architecture, right? Okay. So the latest version of this just came out a few weeks ago. So we went from 2016, 300,000 people to actually 1.1 million people in just 2 years later. So now, this came out in Nature Genetics, we literally have 1,271 independent genome-wide genome-wide significant associations. So that's our Manhattan plot, and again, we see the effect sizes of any one of these SNPs is actually tiny. And we also see that the previous associations that we found, they're not disappearing. They typically just get stronger, right? So they just get lower p-values, as you would expect if they're truly associated. There's a lot of biological annotation that you can do with that. I'm not going to go into that, but the good news is what we find is primarily genes that are expressed in the brain and not in the small toe. So that's that's encouraging. Also, when you do an annotation of when in the life cycle these genes tend to be expressed in the human body, it turns out that in the 2016 paper, the signals that we found there, they were primarily expressed prenatally. So, before babies were born, these genes kicked into effect. In the 2018 paper, which had a lot more genetic signal, the annotation actually seems to be like across the entire lifespan. And so it goes from early prenatal to late adulthood. And there's no significant difference anymore when these genes tend to be expressed. So my interpretation of that finding is that in EA2, we were picking up genes that tended to have slightly larger effects, so we could detect them with a smaller sample. And it turns out, so my interpretation would be that the genes that are most important in terms of their effect size for education attainment, they are the ones that are prenatally expressed, but then ultimately the genetic architecture of the trait is so complex that you literally pick up anything that happens in the brain across the entire lifespan. So this is also true if you just look at what is known about many of the genes that now pop up as being genome-wide significant, and they actually, so what is known at this point, for many of the genes, we actually don't really know what they're doing. But for those that we do, actually just mapping the known biological function of these genes turns out to basically map the entire machinery of the brain. So anything that happens in the brain seems to be to some extent influenced by genetic variation, and we're picking that stuff up with education attainment. So here are the polygenic scores. So we talked about that earlier. So you would expect that the polygenic scores get better as the sample size increases, and that's exactly what we see in practice. So we went from having polygenic scores that capture about 2 or 3% of the variation in years of schooling in EA1 to about 5 or 6% with EA2, and now we're actually up here. So we're now in a range where our polygenic scores, they capture more than 10% of the variation in years of schooling. So that is substantial. This is something that is actually useful for social scientists.
So wait, sorry.
Yes?
It's sample size conditional on some kind of shared social environment.
Yeah, I mean, so what is shared here is that we're primarily looking at individuals that grew up in relatively wealthy Western countries. They're all European ancestry. And they all had a schooling system that was basically accessible to all or most of them. So these are the,
So if you pull in a bunch of additional people from a wide variety of different circumstances, you might expect that to go back.
Oh yeah, so that's totally true. So this is actually, so we see some evidence for that in EA3. So one thing we did is we looked at the genetic correlation between samples. And it turns out that this is far from perfect. So the average genetic correlation across samples is only about 0.8. So that means that although we're looking at samples that were born in the early 20th century, samples that are born later, samples that are born in West Europe versus America versus Australia, and there are clear differences in genetic architecture of education across samples, but there's also a strong shared component which we're picking up here with the GWAS. Also, what is important to know, I touched upon that earlier. So if we take the GWAS results that are here entirely for European populations and we try to predict years of schooling in non-Europeans, the predictive accuracy goes down dramatically. So there is a part of EA3 where we construct this polygenic score in African Americans in the health and retirement study. And what you see here, this is the estimate for the Europeans in HRS, and we're capturing almost 11% of the variation. If we do the same analysis with the African Americans, we're not even predicting 3%. So it's dramatically lower. So you cannot just take the results from Europeans and impose them upon non-Europeans. So that's not how it works. So let's put this into perspective. So the polygenic score, it predicts to some extent, or it captures variation in years of schooling, but it also predicts other things that are related to years of schooling, such as whether people predict complete high school or whether they complete college, grade retention. This is Peabody verbal, this is total GPA, math GPA, science GPA, verbal GPA. So the polygenic score actually predicts these things across the board to some extent. It also predicts total cognition and verbal cognition scores. What it does not predict is variation or decline in total cognition or verbal cognition in elderly. And it also doesn't predict Alzheimer's disease. Interesting. So if you compare the predictive, the amount of variation that is being captured by the polygenic score with other variables that we're typically using as social scientists, turns out the polygenic score now is pretty useful. So if you run a regression of years of schooling and you include parental education, so both mother and father in the regression as a phenotype or as a control variable, that regression, what basically it would capture roughly 20% of the variation. So parental education accounts for about 20% of the variation in the years of schooling of the offspring. If you only take the mother or the father, that becomes a little bit less, but it's still substantially above 10%, maybe 15%. The reason for that is that there is strong assortative mating for education attainment, right? So women have a clear preference for men that are at least as educated as they are themselves, and the other way around. So a strong assortative mating on that. So as a result to that, there is actually not that much additional information in having education measures from both parents. If you control for the verbal cognition score of the child itself, it accounts for about as much as the polygenic score. But the polygenic score actually captures more of the variation than household income, which I find pretty interesting, and clearly also for other variables that we typically include, such as marital status or something like that. Yes.
So how strongly correlated are these? So can you include both of them, or are they super multilinear?
No, they're not strongly correlated with each other. So I mean, if you have a sample of at least 100 people, I wouldn't be worried about multicollinearity at all. So.
So how much would you, of the variation, and would you capture if you included both polygenic scores and the same one?
Right. So there is actually a figure in the paper that shows you how much of the variation can be attributed to the polygenic score if you include all these control variables. So then the polygenic score still captures about 4 or 5% over and above all these other measures. So it's not nothing. Yes.
Maybe going along with that a little bit. So if you're including both parents' education and you're saying that education is highly heritable, how does that work out in terms of, because you share your genes with your parents and you're saying education is heritable, so is there some correlation?
Yeah, there is correlation between these two, but it's far from perfect, partly because parental education is also a factor of their environment. And partly because the polygenic score is noisy, so it doesn't capture the entire heritable component, right? So people have other research groups have used our polygenic scores for a lot of really cool analysis. So this is really getting traction. And I'm just showing you some results here from a paper that Dan Balsky and colleagues have recently published in PNAS. So they use our EA3 score to predict various measures of socioeconomic status in samples that were not included in our study. And the cool thing about this graph here is that it has a population estimate and a within-family estimate. So the within-family estimate is basically using it's using, I think, siblings or dizygotic twins, and just includes family fixed effects in the regression, right? So you see that the education score predicts education, but also occupational status and wealth. And it's even predictive after you control for family fixed effects, which means that the polygenic score is definitely predictive over and above any sort of environmental confound that you may have with genetic variation. Also, that paper showed that across the various samples, you see an effect of that polygenic score with socioeconomic attainment of the children for various strata of socioeconomic status of the parents. So they basically they looked at low, middle, and high socioeconomic status of the parents, and then plotted the polygenic score, which is here on a Z scale, measured against socioeconomic attainment of the children, also measured on a Z scale. So this is really cool. So you see a positive slope of the polygenic score and socioeconomic attainment across the various samples and across the various strata of society. But you also see two other things. You see that the average polygenic score across these strata of society is actually not significantly different from each other, right? So there isn't that much genetic variation between people that were born in low SES families from high SES families. And yet, being a winner of the genetic lottery has an effect in all of these samples, right? So you're better off as a winner of the genetic lottery, independent from whether you're born in a poor or rich family. But then you also see that the environment of the parents has an effect that is over and above, clearly over and above, the genetic endowment of the children. So you see that the overall distribution of socioeconomic attainment shifts upward with the socioeconomic status of the parents, right? So there is clearly also an environmental component. Yes. Yes.
Avoid the reductionist interpretations of these heritability outcomes. And I'm wondering about what we do with these kinds of results in terms of the recommendations that for intervention, right? I mean, when you show effects that say that parental education has a big impact, children's educational attainment in response to that is like, okay, well, let's make sure to target the children of parents with low educational attainment to assure that they have better chances.
Right. Right. So I think, I mean, so this is really a tricky issue, because I think you cannot answer that question without making it first clear what is your philosophical point of view and your political point of view, right? So, so a lot of, so I would argue that, if you're just considering your genetic endowment as something that you cannot influence as a person, I would argue that you could make a pretty strong argument in favor of intervention and redistribution, because clearly the folks that won the lottery, they're doing better in life, partially for reasons that they have absolutely no control over and that they can take no credit for. At the opposite, a lot of the people that are at the bottom end of the distribution, again, they're doing worse in life, not for things that they caused themselves, but they're entirely out of their control. They were endowed with it even before they were born, right? So I think, depending on your philosophical point of view, you can actually take these results and make a pretty strong case in favor of redistribution. But, I mean, yeah.
So, and you think that it's specifically the sort of the genetic pieces of additional information that help make that case?
I think so.
Of the case that you could make for redistribution based on
Yeah.
social inequality?
Yeah, I mean, typically, so typically the arguments that people make in favor of redistribution, they're all related to things where people experience life events that are basically out of their control, right? So as long as you can take credit or control over the outcome, I think it's more difficult to make a case that that you should intervene and that there should be redistribution. It changes the incentives and so on and so forth. But here we're clearly seeing, I mean, this is pretty clear differences in socioeconomic attainment that depend on something that is completely, entirely out of the control of the individual. So I would argue that this is a case for intervention and redistribution. Just a small reminder, so if you have a polygenic score that captures 10% of the variation or more, that variable is going to be useful even in very small samples, right? So you don't need large samples to use the polygenic score. So, so here, for example, if you have a polygenic score that has 10% R-square, you already have 80% power to detect the association of that score with your outcome at the 5% level in a sample of only 75 individuals. So that's a small lab experiment, right? So, if you run an expensive laboratory study where you do brain scans, or you do an expensive policy intervention where the intervention costs thousands and thousands of dollars, please, by all means, collect genetic data on your individuals, because you can construct hundreds of polygenic scores, use them as control variables, and boost the statistical power of identifying the treatment effects that you're actually interested in, right? So this is super cheap, and people should just do it, even if they don't care about genetics at all. Okay, I think we ran out of time, so I'm going to skip the income stuff. I'm just going to give a little shout out. We just started a new large-scale GWAS that is going to look at individual income, well, we have a study design that's going to take various measures of income into account, and it's actually going to use modern techniques for basically modeling and benefiting from the genetic relationships between these various measures of income. We have some preliminary results from the UK Biobank, which are super interesting. So we find a lot of genome-wide significant signal that is actually unique and hasn't been discovered for any other trait before, including education attainment. And this is stuff that we will keep working on in the next couple of years, and I think this will also yield polygenic scores that are going to be useful for a lot of social scientific research. Okay, conclusion. So I'm very bullish about integrating genetics into the social sciences. I think there are huge benefits from doing that. There will ultimately help us to better understand environmental and policy effects. I think for most traits that we care about as social scientists, actually, we're going to care more about the polygenic score than about any specific gene or the biology that is underlying it. And the good news is that with large GWAS samples, we do find replicable signals, and we do have polygenic scores that are now useful for a lot of applications. So I would say social science genomics is ready for prime time. So that's my message to you. Okay.
If you have questions, additional questions, please feel free to come up afterwards and introduce yourself to the phone.
Absolutely.
It was a real pleasure to have you visit us. This is very informative.
Thanks. Okay. See you guys.